starts from bone tissue engineering, stem cell differentiation, breast cancer research, and bone metastasis. He has got numerous awards and achievements. He is a Young Researcher Award from American Society for Bone and Mineral Research, Travel Award from American Society for Bone and Mineral Research. He is a member of Cancer Institute of New Jersey. Then he is adjunct professor in Department of Endocrinology, University of Madras. Again, adjunct professor from Department of Nanoscience and Technology, Tamil Nadu Agriculture University. So to his credit, he has H-index of 54 and citations more than 9,000 citations for his research papers. He is a leading researcher in the field of cancer biology. So now Professor Salumuran sir will take over the chair. Sir.
Good morning to all. Thanks Dr. Devananda for giving introduction about me. So this is the 6th International Conference on Nanoscience and Nanotechnology. So I welcome all participants for attending this meeting. So today, day 2. So this session contains, 3 invited lectures. 3 invited lectures. As Dr. Devanand said, all 3 speakers are from Shizuoka University. So the first speaker is Dr. Meizam Sarwar Jyoti. So let me say a few words about Dr. Meizam. He is currently working in the Department of Molecular Biology, Shizuoka University, Japan. Dr. Meizam is currently doing research in cancer research. He is also working in animal communications and bioinformatics. His group is also currently working on pharmacological effect of Terminalia biliarii. So I welcome Dr. Meizam for the conference. I request him to deliver his talk.
Thank you very much. So, I'm going to start my presentation. This is Mohammad Meizam Sarwar Jyoti, PhD student under supervision Professor Toshinobu Tokumoto, Shizuoka University, Japan. So I'm really honored that I got the chance to be a presenter of this conference on nanoscience and nanotechnology, organized by SRM Institute of Science and Technology from Chennai, India. And also grateful to my professor Toshinobu Tokumoto for his supervision to allow me to do research in such a field. Today, I'm going to introduce the brand new method of newly established binding assay along with the newly categorized membrane receptor alpha protein named membrane progesterone receptor alpha that recently included as a protein and patented to Professor Toshinobu Tokumoto. So here is my title, Establishment of Steroid Binding Assay for Membrane Progesterone Receptor Alpha PAQR7 by using graphene quantum dots. First, we will know about the progesterone, membrane progestin receptors. A group of steroid hormones, progestin, have the variety of function in regulation of reproductive function of the body and act as a key regulating factor in controlling the reproductive tissues. And natural progestin, named progesterone, plays an important role in regulating egg growth and menstrual cycle, along with the sex steroid estrogen in human. And membrane progestin receptor is the first identified membrane steroid receptors that are responsible for mediating the rapid non-genomic action. MPRs are member of a group of cell surface receptor, progestin and adipose receptor, PAQR family, and the seventh gene of PAQR family, MPR alpha, is one of a subtype which have been suggested to be involved in the physiological roles in reproductive tissue. So if we talk about the rapid action of steroid, we have to say that the use of steroid as a rapid relief came away from long ago. For example, the nasal spray used for asthma patient, and the most common steroid named glucocorticoid that used as a nasal spray. And during surgery, also corticosteroid are the frequently used steroid to relieve pain with rapid action. Thus, the pharmaceuticals already using the benefit of steroid with fast onset of action, but the mechanism not yet identified. So, as MPR is world first identified membrane steroid receptor, so the basic mechanism for MPR as a cell surface receptor, I'm describing here in brief. So there are two steroid receptors. One remains in the nucleus of the cell, another one remains in the membrane of the cell. And basically, steroid bind with the steroid receptor. So when steroid bind with the nuclear receptor, the cellular response to activate the second messenger is relatively slower than the steroid bind with the membrane steroid receptor. So already I said in previous slide about the use of steroid as rapid relief. So we are using these advantages by this mechanism, and due to the predicted structure of MPR alpha have 7 transmembrane domain that belongs to structure of GPCR. So GPCR, also known as the G-protein coupled receptor and a 7 transmembrane domain receptor, because here we can see the 7 transmembrane domain that they can pass the cell membrane 7 times. That's why the scientists named the 7 transmembrane domain. And this structure predicted that MPR alpha also have the same type of 7 transmembrane domain. That's why we say it belongs to GPCR. And the binding domain N-terminal that detect the molecule outside of the cell and activate the cellular responses. And due to the largest genes of human genome, around 700 to 1000 genes among 25,000 genes encoded by GPCR, this is the very attractive area for new drug discovery. And due to the unique structure of GPCR, this study is very much interesting, and almost 50% of pharmaceutical focused on GPCR recently to develop various types of drug. Some of drug already established and approved by FDA. You can see the percentage around 34%. And as GPCR is a high demanding field and MPR structure belong to GPCR, the study of MPR is very much interesting, and already we succeeded to identify and produce recombinant MPR alpha by using yeast and successfully purify as a membrane receptor protein. So thus, in our laboratory, we are trying to establish a screening system by different binding assay, cellular level binding assay and in vivo binding assay, already established and patented to our professor Toshinobu Tokumoto. So as I said, recently we succeeded to produce recombinant HMPR alpha protein by using yeast. So I trying to establish high high throughput screening by using HMPR alpha as a molecular level screening system. So based on this hypothesis, we select graphene quantum dot as an effective nanomaterial that able to couple with protein. So here shortly I will talk about the quantum dot, a nanoparticle which have the properties to show fluorescence under a specific wavelength, and the graphene quantum dot are carbon-based nanoscale particles that exhibit the excellent chemical, physical, and biological properties, and that's why now worldwide using in nanomedicine. We also select graphene quantum dots to develop a screening system due to the very easiest way to produce. So here we can see the purified HMPR alpha that biologically active to show the binding activity. I purify this protein to develop the binding assay. Here the CBBR and Western blot indicate the successful purification of HMPR alpha. After that, I also prepared the graphene quantum dot in our laboratory by the pyrolysis of citric acid in solution of 1% sodium hydroxide. Then check by specific wavelength 360 nanometer light, and we can see in this image only GQD able to changes the color in the specific light. By this we confirm the GQD we prepared successfully. Then I go for activation of functional group of GQD by using NHS and carbonamide, hexaxinamide and carbodimide, and coupled with MPR alpha. Here GQD coupled with MPR alpha by after functionalization of the functional group in GQD. I mean, I indicated the carboxyl group of GQD. Then go for checking the successful coupling of GQD MPR alpha. This is the transmission electron microscopy image. So in this image, you can see that the different size of dot included 2 to 14 nanometer in size that suggests the successful preparation of GQD MPR alpha, and the approximate size is calculated 9.4 nanometer. Then we go for FTIR. Here we can see some peak are belong to GQD and GQD MPR alpha. So at 1400 and 1500 per centimeter, that indicate the presence of carbon-carbon double bond stretching and carboxyl group respectively. And a broad group of 3400 confirm the characteristics of hydroxyl group from carboxyl group. Hydroxyl group from the carboxyl group, and after surface functionalization with MPR alpha, we also see the peak attached in 1644 that assigned as the amine group appears with the decreased intensity of 1593. In addition, GQD MPR alpha contain another peak at 1710 per centimeter, which correspond to carbon nitrogen single bond stretching. So this specific strongly suggests that as prepared graphene quantum dot successfully coupled with MPR alpha. So the changes of peak at 1710 and 1644 indicate the carbon nitrogen bond by confirmed the successful coupling of GQD MPR alpha. After successful coupling, I want to say that to make familiar. This is the graphical figure here. This one indicate the MPR alpha, and this one is the binding steroid binding site. After activation of functional group in GQD, then GQD coupled with the charged amino residue of MPR alpha, and this one indicated the FITC, and here we can see the structure indicated the progesterone, and this is indicate the progesterone. Shortly we named P4. To prove binding assay, we used progesterone labeled BSFITC, that progesterone BSA conjugated with fluorescence level isothiocyanate. To prove our binding assay, we used this one. So we expected that by binding of steroid domain with GQD MPR alpha with P4, GQD and FITC come closer, and if we excite GQD by 370 nanometer light, the emission of 470 nanometer will excite this FITC by FRET mechanism. By this, we can detect the progesterone binding. So now I'm moving to talk about the actual data I got. Here in this graph, I tried to select the appropriate wavelength for GQD MPR alpha, that which excitation wavelength show the highest emission. So I put 4 different wavelengths, excitation wavelengths from 350 to 380 nanometer, and scanned the emission wavelength. So we can see the highest emission peak showed at 470 nanometer by the excitation of 370 nanometer. So I select this specific wavelength for our screening system. So here in this graph, this is the scanning data of binding of GQD MPR alpha and progesterone level BSFITC. So this blue color indicated that GQD MPR alpha used as a control, and the red color indicated that GQD MPR alpha along with the progesterone level BSFITC, and this green color indicated the GQD MPR alpha progesterone level BSFITC along with the free progesterone. Here we indicated free progesterone. This means the much amount of progesterone. So by this scanning data, we confirm the binding assay by this quenching, because when GQD MPR alpha bind with progesterone level BSFITC, GQD and FITC come closer, and the FRET from GQD to FITC show the higher intensity. Whether adding of free progesterone in the same reaction mixture, GQD release the progesterone level BSFITC and bind with this free progesterone. Thus, they create a distance between GQD and FITC. Here we can see the difference in these two peaks. This quench confirmed the steroid binding. After that, we go for the checking the specific binding of MPR alpha and progesterone. Here we also prepare the GQD BSA to check the binding assay as a control, and in same way of GQD MPR alpha. Then we check the intensity at 370 to 520 nanometer. Here we can see the difference of these two bars. In case of GQD MPR alpha, here we see the significant difference by the subtracting data of GQD BSA and GQD MPR alpha. So in case of GQD BSA, almost no changes, but in case of GQD MPR, we see the significant difference of subtracting from these two bars. So by this, we confirm our scanning system successfully established. Then we go for the computing binding of various steroid and analogs. So here I used 8 different type of steroid and their analogs. P4 indicate the progesterone, hydroxy P4 indicate the 17 alpha hydroxyprogesterone, mifepristol is an antagonist of MPR, E2 indicate the estradiol, T indicate the testosterone, and methyl T indicate the 17 alpha methyl testosterone, and another steroid cortisol and corticosterone. So among all, we can see that only progesterone and 17 alpha hydroxyprogesterone show the specific binding as MPR reacting steroid. Here we can see the competitive inhibition with different concentration. Here I use the concentration from -4 to -11, and the dose dependent inhibition confirmed the significance of this screening system. Also, no binding of other steroid and its analogous, the confirm the specificity of the steroid and steroid receptor binding assay. So from this result, we can explain the mechanism of this binding assay system. So this is the mechanism of molecular level screening of steroid and steroid receptor binding. I already told that by binding of steroid domain with GQD MPR alpha with P4, GQD and P4 come closer. And if we excite the GQD at 370 nanometer, then GQD release the emission at 470 nanometer and FITC absorb this 470 nanometer by Fred mechanism. And when we add the free progesterone, this one indicating the free progesterone, so in that cases, we can see the binding domain bind with this free progesterone and release the progesterone level BSA FITC. In the meantime, they are created distance and the intensity become decreased by freight. So this indicate the specific binding of steroid and steroid receptor. After that, I want to add something about new compound that act as MPR reacting compound. So Padina pavonica, known as the seaweed, that available in Indian Ocean and Pacific Ocean. So we select this seaweed as an interesting target for natural steroidal compound. This plant continuously secretes the natural compound that predicted as a natural steroidal compound. So we're trying to isolate this steroidal compound secreted from this seaweed that have the vast target for pharmaceuticals, and already in our laboratory purified some of the secreted compound to check the activity as a MPR reacting compound. So here we can see the chromatogram and computative binding of the compound secreted from Padina. And this is the successful separation by HPLC. Here we got 4 fractions that were isolated from Padina pavonica and check the activity by this method. So I use these 4 fractions and check the activity. I use the progesterone as a control, and we can see the successful binding activity here in this graph. So also this result confirmed by radioactive binding assay. Then we check to make them more specific and to emphasize strongly that our binding assay system or our scanning system was okay to use, okay to actual analysis. Then we use this one, and by this we can say this binding assay is applicable for actual analysis because the previously established the radioactive binding assay result and the result we found by this assay is same. That's why we are can strongly say that this assay is applicable as an actual analysis. So finally, I want to add some physiological role of MPR alpha to emphasize its importance. So a lot of MPR mediated non-genomic action of progestins were demonstrated too, such as induction of oocyte maturation, quick activation, signaling of breast cancer cell, mammalian hypermotility, induction of lordosis, female mating acceptance behavior, and particular interest is the action of neurosteroid in the brain and a progestin in brain allopregnolonol, which is attracting attention as a therapeutic drug for Alzheimer's disease. Recently, it was suggested that anti-apoptotic action of allopregnolone on neuronal cells are mediated through MPRS. Since the binding assay system for MPR alpha is expected to be applied for screening of new compounds for pharmaceuticals, we tried to establish the new assay system using recombinant human membrane progesterone receptor alpha protein and graphene quantum dots in this study. Also, I succeeded to establish a new strategy to measure binding activity of the compound against human MPR alpha, and we're hopeful that will be the very much interesting analytical assay for the pharmaceuticals to develop new drug discovery. So we focus on this analysis and already established by this assay. So I want to finish my presentation. So thank you for your concentration. That's all. Please, question and comment.
Yeah, good presentation, Mason. Yeah.
Thank you very much.
Now the session is open for discussion.
Yeah.
Participants can post your question in the chat box. It will be communicated to the speaker.
Yes. Mason,
Yes.
I have a couple of questions. Just
Yeah,
okay.
sure.
How did you purify this membrane progesterone receptors alpha? Which method you follow?
Okay. We produce this HMPR alpha by using yeast. This recombinant protein we produce by yeast. I mean, for large amount of production, you use yeast, and after production, I mean, after expression, the successful protein HMPR alpha, we check by Western blot and go for purification. So here we prepare the vector construction by using histag, polyhistag. So due to adding the polyhistag, we use the nickel chromatography to purify the protein. The first step, we go for the nickel chromatography. Then when we purify by nickel chromatography and check by CBBR and Western blot, the specific molecular weight, we know that this one is around 50 kDa. So after this confirmation, we go for another purification by cellophane amino resin. This also column chromatography. And when we use the cellophane amino resin to purify this HMPR alpha 14, then we this resin almost able to purify this protein. And after that, we concentrated the protein by reduced amount of cellophane amino, and then we check the binding assay that it was really biologically active or not. Then after confirming the binding assay, we say that our protein was successfully purified.
Mason, the question why I asked, in general, we use EcoRay system. In general,
Yes.
we use EcoRay system to,
To recombinate protein.
to clone and, induce and express and purify. Okay. And then why you usually FITC fits to BSA? You can't directly put it into progesterone receptor alpha or steroid. Why you put it to BSA? Can you reason? FITC.
Progesterone level BSF ITC, I mean, why we use this one.
BSA.
So Actually, we first conjugate the that was commercially available, that progesterone level BSA.
Oh, BSA
And
fixed available. Okay.
yes, progesterone level BSA was commercially available, and we buy separately progesterone level BSA and FITC, then conjugated the method instructed in our laboratory. So
Okay.
I can
So,
state the
yeah, I
the
know.
bind.
Yeah, company, they are selling a number of, fixed conjugated stuff. Okay, that's what I want to know. That. Why the BSA they put. Audience, any questions, audience?
I have a small clarification.
Sure.
This tagging of membrane protein, how you did it on graphene quantum dots? Is it by simple incubation or you used any specific method?
Please ask again. I can't got.
You conjugated membrane protein to graphene quantum dots.
Yes.
What method you used in specific?
To conjugate?
Yes.
Okay. So first we dialysis the graphene quantum dot.
What?
The prepared graphene quantum we dialysis by 1 kilodalton membrane. Then this dialyzed graphene quantum dot, I use the N-hydroxysuccinamide and the carbonamide to activate the functional group of graphene quantum dot. We know that graphene quantum dot have the in its airs the carboxyl group and a hydroxyl group appeared. I mean, this functional group activated, and I told that our HMPR alpha have the charged amino residue. So this charged amino residue can able to go for peptidic bond with this carboxyl acid. That was functionalized by NHS and carbonamide of GQD, and by this peptide bond, we conjugated the MPR alpha and graphene quantum dot.
Okay.
And also, yeah, thank you
very
very much.
One more question from Dr. Michael, and yeah, please.
Hello.
Yeah.
Can I ask one question?
Yeah, sure.
Oh, hello. I'm Keita Isago from Shizuoka University. So I have just a basic question. Like in this slide, you showed that the combination of your protein and GQD, all those things, have a better affinity towards the P4. I mean the free P4, but not the bonded one. Why it is like that?
Please again.
I mean your conjugates, the whole conjugates can, attached with the free P4, right? But not the bonded one. I mean, when there is a P4, it can release the bonded one and go to with the free P4. So why the affinity with that bonded? I mean, the free P4 is higher than the normal one.
Because we use the same amount of bonded P4. I mean, we use P4 BSA labeled FITC, the same amount of P4, free P4 we added. That's why the much amount remain in the reaction mixture, and we in our membrane progesterone receptor only one binding site in terminal. So this one release this very small amount of P4 that attached with the BSA FITC and bind the much amount of progesterone, free progesterone. This is the, I mean, I can clear you. Due to adding the much amount of free progesterone, they are generally the affinity rate to bind with this much amount of free progesterone and release the free progesterone labeled BSA FITC. Okay. Hello.
Okay. Okay. Thanks, Mason, for giving a wonderful talk. Next we move on to next speaker.
Yeah, thank you very much.
So next speaker is Professor Toru Aki,
Yes.
Department of Nanovision Technology, Shizuoka University. So Dr. Toru Aki is a professor in the Research Institute of Electronics, Shizuoka University. He is a Chief Technology Officer of ANSI and mainly engaged in CDT-based X-ray gamma imaging systems. His research interests are development of imaging devices, systems, and their applications for invisible light, especially X-ray, gamma ray, neuron from base to practical. So the title of his talk is Photon Charge Counting Type X-ray Imager. Yeah, I request Professor Toru Hauki to be presented.
Okay. Thank you, Chairman, for kind introductions. Okay, so can you see my slide?
Yes, yes.
Yes. Okay. Okay. I'll start my talk. Today, so I'll talk about the photon and charge counting type X-ray images. So as you know, so photon is a countable digital signal. So and charge is also countable. So one unit is just one electron. So we made a fully digital counting type, so X-ray image. So today I will show you some results. So my name is Toru Aoki from Shizuoka University. These are the collaborators, also my collaborators. So including our institute and unseen. So I want to say the special thanks to the Professor Keitaro Hitomi from Tomofuku University. So I use his slides in my talk. Sorry. Okay, this is the outline of my talk. So I will show some introductions at first, and then I will show the photon and signal, photon and charge counting signal processing. So and then I will show some device of the photon charge counting image, cadmium telluride stuck on the silicon lead out IC. So and then I will show the new result by using thallium bromide, one of the X-ray to signal converter materials. So okay. Okay. This is next slide. At first, I will show the introductions. So this is very simple introductions. X-ray imaging is widely used as many applications. So recently, they request more high resolutions, high sensitivity, and so on. Okay, so this view graph shows us our research field. So many areas, but not so wide. So very focused. So but from a material, so crystal growth of the thallium bromide was cadmium telluride, sensor. We made some electrode, some doping, and so on. And another case, we developed the read out IC. In this we use the ASIC process. So and then we combine the sensor and read out IC. So we made the X-ray detector and images. Today, I will show these devices. And signal processing is more important. And X-ray measurement systems, optics system is also. And we developed the original application software and so on. Okay, last time, in ICONN 2019, I showed this kind of the energy response of the attenuation coefficient of the many materials. So by using difference of this energy response curve, I show these kinds of results. So this is calculated results. This is just penetrated image by X-ray. So this is real materials. This is a Japanese chocolate, Japanese caramel, and this is a dangerous materials, plastic boundaries here. So by using penetration image, so we cannot understand which is dangerous and so on. So sometimes we use cross-sectional image by taking by computed tomography. So we can know this kind of the cross-sectional images, but still difficult. But by using energy information of each X-ray photons, we can calculate this kind of two kind of the maps. One is effective atomic number, and one is electron density. So atomic number is almost the same. So average atomic number is almost same. So however, electron density is very high in the dangerous materials. This result is shown in the last time. So today I will show the signal processing method. Usually, conventional type X-ray image are used as scintillator materials. Scintillator is a converter of the X-ray photon to visible photons. So these visible photons. After changing the visible photons, we can use CMOS or CCD sensors like this. So in this case, we can use a very high technological CMOS or CCD sensor in here, as you know. But this visible light is scattered like this. So therefore, some image is not so clear. So, but by using semiconductor, so in my case, cadmium telluride or thallium bromide, X is coming into here. So the charge is directly moved to this direction. So by using electric field. So long time. So our laboratory developed and research and developed the photon counting X-ray detectors. In this case, X-ray photons come into here, so catch here. So and this high energy photon, X-ray photon generates many hole and electron pairs like this. So the number of the hole and electron pair is dependent on the X-ray photon energy, each X-ray photon energy. So by counting this number of the electrons, we can know we can make this kind of plot. So and we can make this kind of energy spectrum. So it is a very interesting method. So however, long time for photon counting, we can make a photon counting. So but charge counting is very difficult. Usually we treated these carriers as current to voltage converter and analog wave shaper. So and then we use AD converter, and then we can get small amount of the information, small information of the just pulse height data. So from these detectors. Okay. So and in my institute, Professor Mimura make this kind of nanovision science. So we have long history of the image engineering. So and recently, many nanotechnology professors coming to here. So and we merge the nanovision science, new image science. It is usually collective statistical treatment of the photons and electrons so long time. So our conventional CMOS CCD almost devices based on this statistical treatment. So but we treat the photons and electrons. So it is individuality. So and electronic handling, photon and electronic handlings in the nanometer region. By using this this technique, we consider new signal processing system. So signal process on the charge domain. This is a detectors. Output signal is the charge. So we develop the charge to digital converter, not ADC, charge to digital converter. So, and so this is so DSP. So this kind of structure, so we suggested. So, and this is real block diagrams here. Detector is here. So the carrier, this charge is moved to the integrator. So integrator has a large capacitance in here. So from here to here, charge is moved to the here. So and this charge is research inject means so here is if here is a hole, so we inject the electron, some hole is cancelled. So by counting the number of the charge injection, so we can digitize by digitize from charge to digital signal, so directly in this block. So by using this circuit, the circuit space, circuit is very small, so it is suitable for the integrated circuit also. So okay, I will show the next slide. So photon and charge counting image by using a cadmium telluride on silicon readout IC stacking structure. So this picture shows a photon counting processor. This is a prototype chip. This is a prototype chip. So in another presentations in my laboratory, so Professor Takagi shows the newest result of our result. They're a little bit different. He shows us more bigger. So, but in my case, it is a first prototype chip is here. So size is a 5 millimeter by 5 millimeter. So, but this area is 3.2 by 3.2 millimeter. So number of pixels is 40 by 40. Therefore, so a pixel size is 80 micrometer by 80 micrometer. It is so small. Maybe now so a CMOS sensor in the smartphone, the pixel size is 5 micron and more less than 5 micron. However, so in the X-ray region, it is so difficult to use the lens optics. So we have to use the real size of pixels. It means if so you want to take my body, so maybe 40 centimeter of the device is needed. So 40 centimeter, 80 micron pitch. It means 5,000 pixels. 5,000 by 5,000 pixels. So it is a very high resolution. Maybe you can understand. So we make these kinds of properties. So this area we have 40 by 40 pixels. So here is expanded to the pixel circuit, analog device and digital is like this. Okay. Okay. By using this device, we can get this kind of energy spectrum. The resolution of the energy threshold is very low. So in our case, so only 16. Usually, 1,000 or 2,000. So you use for the spectrometer. So, but here is it is image. Therefore, the threshold is very rough. However, so we can get another signal. In this case, so we use americium radioisotope. We can obtain here. So and so cobalt 57. So we can get this kind of peaks here.
Sir, you have 5 minutes.
5 minutes. 5 minutes.
Yes.
Okay.
Yes.
Okay. Okay. We can get this kind of out of the value and so on. So okay, we include the digital energy integration. It can obtain the very high intensity in calculation of the high intensity case. So we can get very good linearity. So even with high fluxes. So by using this method, we can get so this kind of good images. Okay, by using this device, we can use not only cadmium telluride but also new material thallium bromide. This is these slides from Professor Hitomi. So usually we use germanium detector and so on. But thallium bromide has very good conditions like this. However, output signal is a little bit different, little bit slow. So usually, we have to make a new LSI ASIC. So however, in our case, in this slide shows us as an example of the thallium bromides. In our case, so our circuit is based on the nanovision science. So we can count photon and charge. Therefore, we can apply this background. So if so, the time response of the detector is a little bit different, but so we can adapt this circuit to the image. Therefore, we make this kind of images. So here is a thallium bromide. Here is our LSS. This is a small bump. It's like this. So this is a real picture. So this is a crystal. So this is a LSI. This brown is a LSS. Okay. This is a result from cadmium telluride. We can make this kind of object. We can get this kind of X-ray penetration image. It is also I showed another conference before. So and this is a thallium bromide case. So this is a background. Photon counting makes no noise background. So but we put the gold wires here. We can get the gold wire images here. So however, we cannot get any image by using character. This is the last character, so in cadmium telluride case. So because thallium bromide has a very high sensitivity in high energy, it is not enough to it means object is so small, so therefore it so thin. Therefore, almost X-ray is penetrated through to the cadmium thallium bromide. So, but by using these kinds of objects, we can get this kinds of images. So we measure some modulation transfer function, so resolution chart. Now, we have the many noises in here. So, but the resolution is so good. So this is nice to use, right? So it is good. So this is a compare chart of the cadmium telluride and the thallium bromide case. So conventional scintillator imager has this kind of resolution. This direction is high resolution. So this value is also MTF value. So we can get this kind of the curve. So thallium bromide and cadmium telluride almost the same curve. So we can get the good resolution images by using the photon and charge counting circuit. So finally, I will summarize this section. So we demonstrate thallium bromide photon counting imaging devices. So this is I think so. It is seem good for the NDT non-disruption testing applications. So we can get good MTF. It means it is high resolution images. So some errors occurred. So but this is a fabrication processes that we have to optimize. Okay. Thank you for attend. I'm sorry. Some. Okay, so, I'm sorry. So thank you for attention.
Thank you, Professor Mori, for giving a nice presentation.
Thank you.
Now the session is open for discussion. I forgot to tell one thing. Professor Tori, his papers have been cited more than 26,000 times.
Sorry.
26,000. his, abbreviations, H index 61. Yeah.
Okay, so please ask again.
Yeah, your award, your achievement. I am talking about your achievement. Yes.
Achievement.
Yes. Your papers, your
Yeah.
papers have
Yes.
been cited. Yeah.
Yeah.
Don't
Okay.
see.
Yeah. What our final purpose is so final purpose of the our development is so making the so large size of the so X-ray images. So by using so this small module, so we will tile the large scales. So in my presentation, so we can make so a good one modules. It's okay.
Yes. Now, audience, yeah, any questions, Now session is open for discussion. So session is open for discussion.
Maybe a little bit different.
Javanan?
Sir,
Yes.
from audience side, there are no questions.
Yeah. Is there any? Okay. If there are no questions from audience, I thank one more time Professor Tori for giving this wonderful talk. On
Okay.
behalf of SRM, we thank you so much.
Yeah, thank you very much.
We move on to next speaker,
Okay.
Dr. Anuhan Datta Sautri. He is from Research Institute of Green Science and Technology, Shizuoka University, Shizuoka. His research interests are, synthesizing different nanomaterials and nanocomposites, their application in sensing and biosensing devices, then their applications in imaging, drug delivery, then super capacitor or energy storage devices. He obtained a number of awards and achievements, for example, JSPS, Japan Postdoctoral Fellowship Award, then MSSIS, Russia Postdoctoral Fellowship, Best Thesis Award from Saga Institute of Nuclear Physics, citations 1063, then H index is 19. So the title of his talk is Detection of Viral RNA by Fluorometric Nanoprobe with DSN enzyme assistance. I welcome and I invite Dr. Anuhan Datta Sautri to deliver his presentation.
Yeah. Thank you, Professor Selvam Murugan. Can you hear me?
Yes, we can hear you. Yes.
Thank you. So can I share my slides now?
Sure. Yeah, go ahead.
Okay. So okay. So now the slides also visible, right?
Yes. Yeah, we can see it, right.
Okay. Okay. Thank you. First of all, I'm really thankful for this platform, because after a long time, I am going to present something, though it is online, but still, actually online presentation is maybe, this is my second time online presentation. I am not very habituated. It's like I'm talking something in the mirror. So whatever I am trying to explain something. So the concept, in this lab in Shizuoka University, I worked here for almost more than 3 years. Now I am actually an assistant professor here. So in this lab, we mainly work on different type of virus sensing methods. Obviously, our main motto is to make some different nanocomposites, and those nanocomposites generally we use for the, or we apply them for the virus sensing protocols. in different approaches also. So recently, I mean, today actually I am going to present something about the fluorometric some nanoprobe for the DNA, I mean the DNA or RNA detection through the DSN, I mean the duplex specific nuclease. This is a specific enzyme for the RNA sensing technology. So first thing, I mean, we no need to tell anything about the virus, especially during this pandemic situation. So I just use some old data that still the virus is a very huge problem. Not only the corona or this COVID-19. I mean, virus is a dangerous throughout the human civilizations. And I just use one quote from a Nature Reviews that if all the 1 into 10 to the power 31 viruses on the earth, if we just one by one like this, so it can stretch for 100 million light years like this. So I mean, there is a huge number of viruses. So this type of outbreaks can anytime be possible. And I mean, we have to prepare for any type of mutations or any type of virus involvement. So that's the main thing. Not only corona, this is the common for any type of virus outbreaks. So the question definitely remains that how to prevent this virus outbreak. The simple concept is maybe we can definitely I mean have some vaccination process, but still, I mean, in this corona time we also know that the vaccine is not a very easy thing. Just after a virus outbreak or pandemic situation, we can't get anything. So we need some protocol, we have some protocol, we need a long time for that things. Another thing is that like the diagnosis. So yeah, definitely if we diagnosed any viral fever or any other fever very quickly, the initial stage, so definitely in that case we can start the treatment process or something in early stage. But the problem of most of the viruses are the symptoms are also very common. So that's why to detect or identify, especially like in India, dengue. I mean, eastern region of India. Actually, I belong from the Calcutta, so I know that the dengue is that kind of disease. I mean, if we can't detect the dengue initial stage, then sometimes it is too late for the treatment process. And without the confirmation of the dengue, we can't start the treatment also, because we all know that there is some problem with some specific medicines with dengue, and sometimes the dengue symptoms are also very similar with the heart disease. So that is the another problem. So early diagnosis is very important. Okay. So in that case, so for the diagnosis system, actually I belong to that type of research. We have different type of method. I mean the target. First, we can directly target the virus, or maybe the genome, I mean the inside the nucleic acid or the protein something. Or another method is indirect. I mean, after the viral infection, maybe any human body can produce some antibodies. So if we can I mean identify the antibodies, that is also confirmed that this man has that viral infection or not, like this. So this is the indirect method. Everything has some pros and cons, obviously, but generally we depend on this type of indirect method, where a lot of negative, I mean false negative or false positive result is also very common. So that's why we are looking for any direct methods. So in case of direct methods, still we have some problem, that is the expensive things are obviously there, and sometimes sensitivity is also questions. And this is a complicated process. We need some expert I mean, human being or something like that. So for these things, I also show something why the direct method is also important. Because like this is a figure of dengue, where we can see the initial portion. This time actually viral particle or the proteins are very high, but not the antibody. After 6 or 7 days, excuse me. So after 6 and 7 days, then we have some increased amount of sorry, this is choked. So after 6 and 7 days, then the concentration of the antibodies is going high. So for if we really want to detect something initially, So then the virus particles or the NS1, the direct protein or surface protein, this is the most authentic way. And sometimes like the virus also have some serotype problem, like similar type of virus, but the stain are different or some nominal mutations are there. Like dengue also have some 4 serotypes. So sometimes this serotype detection is also very important. So in that case, we generally use some different type of method. This is the existing methods, but among this generally we use the most useful techniques, that is the ELISA or the RT-PCR like this. So for a relatively inexpensive, or rapid method, we need some kits, some viral kits. So in that case, we can immediately detect the virus concentration or the response also. Like this is the virus. In this sample, the virus is yes or no, like that. But we know, everybody, we realize that there is a severe problem of rapid detection kits, that is the reputation or the reliability. That is still, maybe if you search in a Google Scholar or Web of Science about the quantity of the paper of the biosensor, you can find a lot of millions of paper on maybe glucose sensor or something, but still the reliability is the questionable things. So that's the thing. We need to do something more reliable technique to measure this virus or this type of analytes. So in that case, we generally use the nanomaterials. In our lab, we also use different type of nanomaterials like gold, carbon, ZQDs, quantum dots of like this, and use their optical or the electronic or electrochemical properties for the virus detection technologies. So sometimes for the complicated or some very sensitive way or sensitive things, we also use some very advanced type of nanomaterials, where not a single component, but a complex of different component mixed with a specific structure, a specific shape, and that particle or that nano composite can be used for the high level sensitivity. So now the topic of the virus detection, there is a 3 type of targets, easy targets. The first one is the surface protein or the direct virus. The next one is the nucleoprotein. We just destroy the virus and target the nucleoprotein inside the virus and also detect that one. So, and the last one or the most authentic one is the RNA or the DNA inside that virus, because some virus have RNA, some virus also has the DNA. So if we can detect the inside the RNA molecules, this is the most stable form of detection, because as the surface of any virus, like even in case of corona also, they mutate very rapidly. So if we can make a stable detection kit for any particular surface protein, maybe after some time it can mutate. So that's why if we can target the nucleic acid of any virus, so that may be a very stable process. Stable detection method. But there is also some problem, like in case of nucleic acid or RNA or DNA, we need some extraction process or the pre-treatment process. So sometimes the sensitivity is questionable. So that's why we use the RT-PCR type of things, where we can amplify the initial RNA concentration. So here actually still, we still, in case of real virus detection, we still rely on the most authentic process of ELISA or the RT-PCR. So in that case, we also use different type of methods, where the colorimetric or the optical detection or the fluorometrics, these are the most convenient one. Sometimes we also use a different combination of those methods for most reliable or more reliable data. So in this case, we use some different type of biosensor. Here, I just want to just take a small glimpse about a colorimetric method. This is also developed in our lab. So in that case, we just try to upgrade our ELISA technique. So in that case, we just make a ELISA type of platform. Here, we use some specific antibodies. After that, we combine the virus, the whole virus particles there. Then here we use some catalyst. Like here, we use the gold and coated with the silver shell, one enzyme catalyst, and then this catalyst can accelerate the TMB reaction. even this is just an upgradation of the common ELISA technique, and we found some better results than the ELISA also. So I mean, in this case, we have a better sensitivity compared to the normal HRP based indirect ELISA. But obviously there is some other problem, like when we compare the data with our RT-PCR, there is some little fluctuation. I mean, this type of data is the ultimate goal of our studies, that we just, after different type of virus analysis, we want to compare our virus data with other, I mean, with the PCR, the RT-PCR, which is the gold standard technique for the viral samples. And then we compare our data with them and try to check that our efficiency of this method. I mean, in some portions, these are okay, but not in a, I mean, perfectly one. And it's mostly in the very low level concentration of the virus, it is not so good. So in that case, we then switch to some other optical detection, like the LSPR. So LSPR is a very common or very well established technique for the optical detection. I mean, not only the fluorescence technique, we just use some extra additional I mean, additional things to enhance the fluorescence signal of a molecule. So in that case, we use I mean, just for a easy understanding, I used a simple technique of quantum dots, some inorganic quantum dots, and a gold nanoparticle. So here, gold nanoparticle can easily give some enhancement or the quenching of the quantum dot depending on the distance. So like if it is a very close, it shows some quenching effect. If it is a moderately distant, then it has a very good enhancement property. And with a very huge distance, maybe there is no LSPR effect. So this is the common concept of LSPR. So the surface plasmon peak of, I mean, electron can enhance or the decrease the fluorescence and intensity of the quantum dot. So this is the common concept of LSPR. So based on this technique, first we try to detect the RNA of dengue RNA. So this is the, so for initial study, we just try to check the synthetic RNA of dengue. We just copy the sequence and make the synthetic RNA in lab, and then we try to detect the synthetic RNA. If the method is working, then we proceed to the real samples also. I mean, real RNA, extracted real RNA. So in obviously, I have to mention that there is some types, serotypes, like 4 serotypes of the dengue. So and this is one paper cutting from a Calcutta newspaper, where the different serotypes are very important factor for the dengue disease. Because in case of first serotype, and then, I mean, after getting the first serotype attack, if you have a second serotype attack, then the, I mean, the fatality rate may be increased a lot. So in that case, we try to check that our method can particularly, I mean, identify the serotype of the dengue, I mean, can or not. So in the sensing mechanism, it's like that. We first use some gold nanoparticle with a small sequence. This sequence actually is completely complementary with this constant sequences of a probe. So this probe, I mean, we use 2 type of probe. Like one is for dengue 1 and dengue 3, and another type of probe is for dengue 3 and dengue 2 and dengue 4. So in this type of probe, the quantum dot is this side, and the another type of quantum dot is the opposite side. So what happened when we already inject or we add the sample RNA, the target RNA. So this target RNA easily can open up the loop formation of this probe, and then it can make a straight type of duplex. So in the one probe, I mean, in one type of probe, the quantum dot can be placed in the exactly opposite side of the gold nanoparticle. But in case of the another type of probe, the quantum dot is very close to the gold nanoparticle. So simply in case of dengue 1 or dengue 3, there is a huge distance like 14 nanometer. This is an estimated distance. And in another 2 types, the closeness is very small, like a 3 nanometer type. So according to the LSPR concept, we expect some quenching on this close formation and very good enhancement in the distant formation. So for the probe mechanism, to generate the probe, we also use some simulation type of softwares to make the exactly round structure. So after that, this is the sensing result. We use some different known concentration of the target dengue 1, dengue 2, dengue 3, and dengue 4. And we can see there is some good enhancement and good quenching in case of dengue 1, 3 and dengue 2, 4. And we can also make some calibration line with that. You can see there is a clear enhancement in case of dengue 1 and dengue 3, and the other two, the dengue 4 and dengue 2, there is a linear quenching effect. So this one, in then we can use a different type of probe. I mean, dengue 1 probe, dengue 2 probe, and dengue 3 probe, dengue 4 probe, like this, and use a different type of analyte inside that probe. Like in this type of structure, we use different probe for different analyte, and we have a very good results. So this is called the selectivity result. So only the dengue probe 1 is only very much selected for the dengue 1 RNA, not in others. The similar trends also found for the another, all the RNAs also. So this is a very nice one. But the problem, not the problem, but the selectivity or the sensitivity of this technique is not as good when we tried it for the real samples of the dengue. So in that case, the intensity is a little bit decreased, and the LOD, the limit of detection, is also a little bit higher. So because in case of the real samples, the RNA is very big, and the second thing, it also contains, after the extraction, it still contains some other things. So the sensitivity is a little bit compromised, but still the principle is working in that case. So that's why, I mean, we conclude from our initial studies that for the low level concentration, or the if we need some reliable and very good reliable data on the low level concentration of the virus, then maybe we still have to go for the RT-PCR. This is the only way. So in that case, I mean, in the RT-PCR, the mechanism is the simple, that they first use the extracted RNA, and then it can cyclic way, it can amplify the main sequence, and then it can detect, or it can give some band or some signals according to that. So just similarly, we also use the same thing with the enzyme that is called the duplex specific nuclease enzyme, that it can also amplify the sequence of RNA without destroying it. So this is the main concept of the current study, that we initially take one RNA sample, and that sample can be amplified to a huge amount of small DNA or small amount of probes. I mean, in that case, the goal is one RNA equals not equals to the one signal probe. It should be one RNA equals to many signal probes. So that is the amplification. So this is the target. I will explain the mechanism later. So initially, actually, there is some paper already published in recent science about the DSN activity, duplex specific. I mean, this enzyme actually extracted from a crab. It has a specific role that it can only destroy or cut the duplex structure when there is a DNA. I mean, if there is a duplex of RNA, there is, no problem. I mean, DSN is not activated. If there is a duplex of RNA and DNA, it can only destroy the DNA, not the RNA. If there is anything like DNA, it can also destroy both the DNA. So the duplex specific nuclease is only activated in case of DNA when it is in a duplex formation. If there is a single stranded DNA, it is not activated. So this is the only concept. So from this concept, already there is a few papers for the cyclation process, but most of the papers are for the microRNAs. This is easy to control, and they also use some basic things like gold nanoparticle, and use the freight mechanism for that one. So in my experience, we also work on gold nanoparticles. So we know that for the real samples, the gold nanoparticle is not a good choice, because it has a lot of thiol interaction. So it is better to either avoid the gold, or it may be coated with something for the real application. So just inspired from this previous study, we also developed our new strategy with the DSN assisted cyclic mechanism. So in that case, we use some tempo. Tempo is also very common quencher of any kind of fluorescence. So here we use one tempo coated gold nanoparticle here. So after that, we attach one DNA, the single stranded DNA here. And another end of that single stranded DNA, the one fluorescent probe, here it is a CDSC, the inorganic quantum dot. Is a very simple things. Maybe in future we can also replace the quantum dot to in another type of probe also, some organic dye or anything. So in this probe, we can use some big size of gold nanoparticle coated with tempo, and it is attached with a probe DNA, single stranded probe DNA, and another end is some quantum dot. So in that case, if after that we add the RNA, so that RNA can bind and form the duplex form here. After that, we add the DSN also. So now the DSN can just cut the only the DNA, not the RNA. So if the DNA is gone here, so the RNA is now free, and this RNA can again attach to the next free DNA in that same nanocomposite, and the free quantum dot can be released from the probe. So initially this quantum dot is very close to the tempo coated gold nanoparticle, so there is a quenched of the fluorescence signal of CDSC. So but after the DSN addition, so it can break the the CDSC and the quantum CDSC and the gold tempo, and it can be free from the tempo surface. So it resulting the good enhancement of the fluorescence. So after that, we just initially characterized with some tame image, so that after the combination of the gold and gold coated tempo with the quantum dots, there is a very close combination like this one. And then we also optimize the each of concentration of the tempo or the gold nanoparticle or the DNA or something. Then we make the whole system for the RNA analysis. So initially we also just test the activity of the DSN enzyme through the UV absorbance. Initially we take the RNA samples and check the absorbance at 260, because 260 is specific for the nucleic acid absorbance. So after that, we also use the quantum dot DNA sequence and the quantum dot tempo also. The similar results are observed. But when we add the RNA, it is little increase when we add the RNA with DSN. But after some time, like 1 minute and then 10 minutes, it's really high. So it means the high absorber means the RNA is already clipped. So that's why the absorbance is going up. But after 10 minutes, there is no significant increasement. And then when to stop the DSN activity, generally we use the EDTA solution. So in that case, we optimize the time also. Like in 10 minutes time, we get the perfect, this is 10 minutes, the perfect time for the DSN activity to complete for a RNA analysis. So after that, this mechanism is followed by the fluorescence study, where initially we use some quantum dots. So after that, quantum dot and tempo conjugation is quenched down to a very, almost a 3 times decrease. So then we add the RNA. Still, there is not a, good enhancement. But after the DSN addition, we can for 10 minutes and more than 10 minutes, we have a very good enhancement of that. So this proves that our proposed mechanism actually working. So after that, we also optimize the concentration of the DSN enzyme with some different of like 0.04 unit of DSN to 1 unit of DSN also. And this sudden change, this change is also, is satisfactory for this experiment. So we don't want to use a lot of DSN for this analysis. So that's why we choose the 0.1 unit of DSN for the further studies. So after that, just for the confirmation or the more confirmative analysis, we use one, like here we use a corona full length RNA, and then we just choose 4 different domains of that RNA, just arbitrarily 4 different domains, and we make from this, with the complementary of this domain, we make the loop formation of 4. and there is 4 type of loop formation here. So this is actually the probe. In this case, we use a loop, or this is called the molecular beacon, this type of structure, and then we use for the analysis. So in that case, we also, make this constant region of this loop formation with AU sequence, because it should be like RNA type of sequence, that the DSN should not break this loop formation. So that's why you use the AU for this bond. So after that, when we add this one to the RNA structure, the normal RNA, it can open and contact, make a duplex formation of that RNA. And after the addition of the DSN enzyme, it can cut and release a lot of free quantum dots in the solution. So we can have a high concentration of the quantum dots in the solution. So in that case, after 10 minutes, after addition of DSN, so after 10 minutes, we add the EDTA to stop the reaction. So from that thing, we also analyze the different concentration of the RNA. Here we try to 1 femtomolar to the 10 picomolar of the RNA concentration. So after that, we also check the selectivity with the different other RNA, like non-complementary or the single mutate RNA. And we also find that our RNA of the 1 picomolar RNA is quite high, and it's get a quite high fluorescence enhancements compared to the others. And the last, we also use the real extracted RNA for that one, but still the data is not satisfactory yet. So we have to think about how to change the probe or the fluorescence probe to make a clear signal for the real sample, the real RNA also. So that is the conclusion of our, my study on these things. We are still in a struggle of the reliability. Sometimes we can find a very good sensitive results, but if that result is not a very specific to a particular RNA or a particular virus, or especially in case of lower concentration of the analyte, sometimes the data is not exactly reproducible. So we are still struggling on the sensitivity. So, but the initial, for these initial studies, we can see, we can confirm that the overall technique is working. Maybe very not good in the very low concentration, which is our main target. But if we can perfectly optimize everything with this LSPR system and the DSN assisted cyclic amplification, maybe in future we can have a very good alternative for the RT-PCR. But now at this present scenario, it is too early to state this type of information, but still we are trying to make something on that. So still we need to do a lot of experiment to confirm the whole things. So thank you for the listening, and obviously I need to acknowledge my professor here. Professor Park is he's also one speaker of this conference. So he's our supervisor, and we belongs to a nano bio group. Here is other members in there. So this is a completely international lab. So we have a lot of Japanese and non-Japanese from different region, and we work in a nano bio group here. So, and JSPS and Japanese AMET projects, we also thankful for that. So thank you all for listening. So please, I will be very happy if someone can ask any question regarding these things.
Thanks a lot, Dr. Rankan, for giving a nice presentation. Now the session is open for discussion. Audience? See, remember, because Dr. Rankan, he already told, this online, mode of presentation, talking to mirror. So audience, please ask question, ask question. I agree, Dr. Rankan, sometimes I also feel, you're talking to wall, talking to wall.
Actually, this is not, I mean, we really don't have any type of experience about this. Without direct interaction, without the eye contact, I really can't enjoy my talk. So that's the thing.
Ankan, I have I just need some clarification.
Yeah,
Some,
sure, sure.
coming to virus detection, mutation of the virus, specifically in case of RNA virus, plays a very important role. And
Yes.
as you have mentioned, when the virus is struggling to replicate inside a host cell or body, then in a low concentration of the nucleic acid of the virus, then your method, now it's not so specific or in some sense. Am I right?
Hmm.
Okay. Then in such cases, when it comes to mutated virus, will this method suit good for when there is a huge spread of virus, called in case of COVID or some situation where you have a sudden outbreak of virus?
Um, yeah, I mean, definitely there is a lot of drawback. I have to admit, but I think, like, still, if we have some problem, I mean, if some new virus is outbreak, and the simple technique is we have to first uh, decode the DNA. After that, we can make some probes and then go for the RT-virus PCR. So this is the only way. So I am also thinking this type of thing. I mean, if our probe and this mechanism is okay for the amplification technique, maybe we can also try for the same thing in the lab. And in case of like a mutation, as you said, I mean, at least the surface protein mutation has a higher possibility than the nucleic acid mutations. So in that case, we try to target on the RNA of the virus. So this is the thing. I'm not sure that I can answer you the exact what you ask.
No, I got your answer. You need a lot of optimization in regarding to coming to exact application.
Yeah. I have to admit that to challenge a very well established method like ELISA or something, or RT-PCR also, is not an easy job. So I can't say anything like this, but I mean, this is a long, I mean, I am in a research career, maybe almost like 9 to 10 years. But from my initial study for the biosensor devices, my dream was like that, maybe I can challenge the RT-PCR or not. I mean, or maybe some footstep or some assistance toward the artificial technique. So that's why I think that this type of amplification technique maybe can help if a proper optimization or the proper nano composite we can develop. So this is the my thinking.
The major output of such devices comes from the tagging of DNA or RNA molecules to the surface of any cadmium selenide or telluride like particles or even gold.
Yeah.
I think that if that is not properly stabilized on the surface, that may have an impact on the output. Now, this is what I feel.
Obvious.
Not only in your system, but any system, that stabilization is not proper or reproducible. Maybe that may lead to a lot of error output when it comes to
Exactly.
practice.
No, actually, I mean, initially we use this gold and the CDS and all those things because of the step. I mean, we already have a lot of data on this type of molecule, like gold and CDS. This is already well established. If our method is, and I want to prove that my method is working or not from these well established things. If everything is fine, then slowly or step by step, I just remove the gold nanoparticle with some other nanomaterials, and then the CDSC with some other, and try to make more stable system for these things. So this is also in my mind, but it depends on the, I mean, each step and the success of each step like this.
Thank you so much. Very
Thank you.
interesting.
Dr. Icon, I have a
Yes.
couple of questions. The first
Yeah.
thing, you mentioned the DSN, duplex specific nucleus.
Mhm.
I think that is similar to S1 nucleus. S1 nucleus. You heard about S1 nucleus?
Um, Sir, I am a chemist. I really
Okay.
don't know
Yeah,
anything.
I valid, because S1 nucleus also it cleaves, it recognizes only double strand RNA.
Double
You
strand
know.
RNA or DNA?
Yes, double strand RNA, only RNA, not
Is it?
DNA. Yes.
Okay.
R, RNA, DNA. So wherever there is double strand, that is protected.
Okay.
Any loop formation, is there any loop? No complementary. That
Yes.
will be cleaved by this S1 nucleus. So
Wow.
that is the one point. Another point,
Mhm.
this DSN, similar to that, the Tuckman, Tuckman probe used in creatine PCR, You know the Tachman DNA probe. You know, there are two types, you know, cyber green and Tachman,
Side 3, side like the.
Tachman DNA probe. You know,
Oh,
rate in PCR.
yes.
It's similar to that, you know, that primer, you know, the probe binds to that specific DNA or RNA, you know, where in polymerase, you know, it extends, then it cleaves that you know that fluorescent probe, you know, that will be released.
Yeah. I mean, we can use the same type of probes in this type of mechanism, right?
Yeah, same thing. Yeah, DSN, you know, DSN nucleus. That's what, you know. So this is commercially available, DSN.
Yes. Now it is commercially available.
Commercially available. Okay,
Yes.
good.
But thank you for your suggestion. I will definitely search about these things.
Look, yeah, so nucleus.
Yeah, sure.
Any questions? Any comments?
Questions from the audience?
Yes. Please unmute your audio, Dr. Mangamma. Unmute your audio. Yeah.
This is regarding your thermodynamically for all this sensing of the viruses. No, I feel it is more important to see the kind of bonding you are having.
Okay.
So in order to know the bond strength, if we know about the bond strength, we can use any spectroscopy like Raman or IR, and you can always confirm the which one is more probable for which virus. So that way, I think selectivity can be improved at the spectroscopic level. That's what I feel. Because you have 3 different viruses and you want to improve the selectivity and the sensitivity. So along with all your studies, I feel the bond strength is kind of strain between the virus and the kind of you are having a sample that can at least help you to improve your sensitivity and selectivity.
I think you are from a physics background.
Yes.
Actually, yeah, I never think like this. What you said, like that, through the FTR or the Raman spectrum, maybe we can improve the selectivity. It's a nice idea. I don't know about these things
Because you can do it in ppm level,
okay.
these kind of things.
Okay. Yeah, maybe I have to read and I have to try also, because generally we use for selectivity the antibodies like monoclonal antibodies, and that is very specific for any type of virus. So only I have this information from any biologist and I use that thing. That is
Yes.
my knowledge. Okay, but
Even NMR will do for you.
Yeah, I can understand.
Magnetic resonance.
Yeah, sure. Yeah, whenever I know, I am chemist, so that's why.
No,
Sure.
chemists, in fact, they are very good in NMR.
Yeah, sure. That's why. Okay.
That's all. Thank you. Nice talk.
So if there are no more questions from audience, then I think I can conclude the session.
Yeah. Thank you, sir. Thank you. I am now just unmute myself. Mute myself. Okay.
No, that's fine, Dr. Rangan. You can, yeah, there's no problem. Anyway, we are going to conclude.
Okay.
So this session, we had, 3 invited lectures given by Mr. Maizam, then Professor Toro Hauki, then Dr. Angan Tata Sautri, all from, Shisoka University. So we had a nice, wonderful presentation. I strongly believe, we gained some knowledge. I'm sure I gained some knowledge, I learned something. Hopefully you also gained some knowledge. So one more time, I thank everyone for attending this session. Thank you so much. See you next time. Bye, Devan.
Thank you, sir.
Yeah.
On behalf of ICONN-2021 Department of Physics and Nanotechnology, I thank all the 3 invited speakers, and I thank our chairperson, Professor N. Selvamurugan sir, for coming all the way to chair the session and giving his wonderful remarks to everyone, and for his very fruitful and knowledgeable interaction with the speakers. Thank you so much, sir. Thank you so much.
Okay, bye.
Thank you. The audience can join in the next session. Thank you.